Hey y'all, it's Ryan, and today I want to talk to you about some really cool psychoneuroimmunology methods. Specifically, how scientists study the process of wound healing in response to a skin injury in the lab using blister wounds. And this is an extension of a previous video that I did about hostile marital interactions in wound healing, but I didn't get to go into as much depth as I wanted to in that video on what I think is one of the most inventive and important ways to study this super relevant immunological process, wound healing, right as it happens. Drop any questions in the comments and I will be happy to answer them. Wound healing in response to a skin injury can also be called cutaneous wound healing. So first I'll give you an overview of cutaneous wound healing itself and focus mostly on the earlier stages before describing how scientists can leverage super cool methods to study psychological stress and cutaneous wound healing in the lab. Okay, so let's dive in. So when we get a skin injury, there's this precise set of processes that help the skin regain both its physical and functional integrity. These are continuous stages that comprise cutaneous wound healing. They're distinct but overlap temporally, that means over time, and include clot formation, inflammation, cellular migration and proliferation, wound contraction, and remodeling. So each of these processes is affected by how deep the wound is and how much tissue the person has lost. Okay, so you've gotten a cut on your skin. What happens next? First, and maybe most importantly, blood begins to coagulate at the wound site. And this means that it's changing from a liquid to form a blood clot. This prevents any more blood loss and also protects the wound from any other junk that might be around in the environment. I won't go into too much detail about each step here because I want to get to how we actually measure these processes, but know that each step has a lot of important immune players, like platelet-derived growth factor, which helps to direct neutrophils and monocytes to initiate the inflammatory phase. The inflammatory phase largely functions to attract, you guessed it, inflammatory cells to the site of the wound. And now remember from previous videos the differences between acute and chronic inflammation. So here we're talking about acute inflammation, which is essential for this phase of wound healing. At the beginning of the inflammatory phase, there are lots of neutrophils involved, but late inflammation is typically characterized by large numbers of macrophages and lymphocytes. So normally neutrophils just help to remove the bacteria that might be contaminating the wound, and then if there isn't an infection, the neutrophils will just undergo apoptosis, so programmed cell death, before being eaten up by macrophages through phagocytosis. And this is really important because acute wounds might not heal if they have a bacterial imbalance, so neutrophils helping to clean out that wound site is critical. But this whole response is quite beautiful, with different immune cells being attracted to the wound at the right time to make a difference. Because although neutrophils might have a limited role in wound healing when there isn't an infection, macrophages and lymphocytes are critical for wound healing. And by 48 to 96 hours after the wound, and in response to the cells of the cytokines and the growth factors released by neutrophils, monocytes flock to the wound and accumulate before being activated and differentiating into macrophages based on the wound environment. These macrophages continue to help clean out the wound from bacteria, remove the neutrophils that went through apoptosis, and any fragments of damaged tissue. This process is called debridement. And in addition to debridement, macrophages are also the primary source of cytokines. Again, a favorite topic on this channel. And cytokines play a huge role in the regulation of wound healing. So hint, this is one reason why psychological stress can slow down wound healing. Now, I'm going to go through this next part really fast because it is super complex and not really the main focus for today. So after an overlapping with this inflammatory stage is the reepithelialization phase, where epithelial cells migrate from the edge of the epidermal wound. There's also granulation tissue, which has a high density of blood vessels. Angiogenesis, or the formation of new blood cells, is essential to give a blood supply to this healing wound and deliver essential energy-providing factors to those highly metabolic cells. So this is also why malnutrition can delay the wound healing process because it is so metabolically costly. The wound also has to contract to help facilitate wound closure. And this happens when special cells called myofibroblasts exert tension on the granulation tissue. And lastly, for real this time, the wound matrix has to remodel. And this stage can last for months or years after the initial injury. Okay, so that's a really quick overview of the wound healing process, which again has these distinct stages that still overlap in time. So what affects wound healing and how do we actually measure it? I'm going to talk about systemic factors today primarily, and not clinician-induced factors like the medications or the dressings on the wound, because that's not what I'm usually most interested in personally. So really quickly, there are also local factors that include something at the actual wound site, like the presence of bacteria, which can result in infection and complicate the wound healing process. As psychologists, we're usually most interested in the systemic factors though. So for example, malnutrition, aging, and psychological stress are all systemic factors that affect wound healing that psychologists might want to isolate and study. So you know which one I'm most interested in, right? Psychological stress, of course. So in a mechanistic study run similarly to the hostile marital interaction study I discussed in that earlier video, researchers gave 36 women each 8 suction blister wounds on their forearms. Okay, so this is the main focus of the video. Blister wounds are so freaking cool because they let researchers actually measure the effects of stress on cytokine production at the wound site. So I'll show you a picture here. So fast forward for about 10 seconds if you don't want to see what blister wounds looked like. So here you can see that there are these 8 sort of suction blister wound areas where they have it on the person's arm. And that's what they do for a lot of studies like this. So I wanted to give you sort of an example of what this looked like. It's luckily not a super gory version, but this is sort of what it looks like in the lab. So for example, in this early study, women who reported higher perceived stress had lower levels of 2 important cytokines at their actual wound sites. And the thought is that psychological stress affects the local wound environment by modulating pro-inflammatory cytokine production. So in addition to measuring locally produced cytokines, researchers want to understand which other factors might be mediating. So explaining this, so researchers often measure salivary cortisol to study whether stress-induced activation of the hypothalamic pituitary adrenal axis is affecting this local immune response. Okay, the last super juicy example that I want to get to you, just to make you want to study this kind of stuff, is an example of a mucosal wound healing study, where the researchers gave 11 dental students 2 punch biopsy wounds on the hard palate, so like the top of their mouth. And first they gave the dental students a wound during the summer, so not a stressful time generally, right? And the second wound was about 3 days before the students' first big exam, probably a very stressful time. And on average, they found that the students' wounds actually healed 40% slower during that stressful time, that stressful exam time, compared to when the students were less stressed during the summer. 40% slower! The key to these kinds of studies is consistency and ingenuity for the comparison. So here they're often creating multiple blister wounds to measure healing in each one. So even when the sample sizes are small, the information can be really invaluable because you have 8 wounds on each person and they likely have multiple daily or weekly measurements depending on the intensity and the goal of the study. So you can assess out a lot of these intermediate processes because of these kinds of studies, which is super cool. Okay, y'all, that's all I have for today. I hope you enjoyed this first methods discussion. Definitely feel free to ask any questions and let me know what you think of this sort of format and if you want more method stuff. And like and subscribe for more content like this.